So today we're going to learn about how to call genes. And essentially what calling a gene means is identifying a region in the genome that is predicted to be a gene, identifying the gene start, which is basically the coordinates of the translational start, and doing the best to predict what a potential function for that gene is. This is output for DNA master and is the frames function. And what we're seeing here are the three possible frames in the genome SANSA. And these top three frames would be forward open reading frames, and these bottom three frames here would be reverse open reading frames. And the long lines that extend top to bottom in a frame indicate stops. And these lines that are midway across the frame indicate potential start codons. So the blue block or bar here is indicating the coordinates for a gene predicted by either both Glimmer and GMARK or one or the other. And this is the first gene predicted in the genome SANSA. Now, if you look at GP4 over here, what we notice is here's the stop for GP4. This is a forward gene. And the open reading frame for gene 4 has 1, 2, 3, 4 potential starts. And it looks like the computer or DNA master has decided that the second start is the most likely start. Our job is to see if we agree with GMARK and or Glimmer, or whether or not possibly this start over here is a better option for GP4. So the other thing we'll determine is what does the protein encoded by this gene possibly do? And the other thing we'll do is look at large spaces between genes and decide whether or not do we need to extend the start of a gene, or did GMARK and Glimmer outright miss calling a gene, and can we identify a gene in that region? So here's an example in the SANSA genome where GP59 is a reverse gene and GP60 is a forward gene, and then there's this huge gap in between GP60 and 59. Now, are there regulatory sequences in here, and so there's no place for a gene, or did the computer program miss perhaps calling a gene here or a gene here? And so that's something that we have to determine. Now, these three genes here are all forward genes, and these genes over here are all reverse genes. And so this is a region of divergent transcription, and we know that in order for transcription to occur, we need promoters. So in a region of divergent transcription, we know there has to be enough space in here for two promoters, one for these genes and one for these genes. So this is a place where actually it makes sense for there to be a large space. Here's the second region in SANSA genome between GP37 and 38, where there's a large gap. And it's possible that there's a promoter here for GP37, but it's also possible, if you look at this open reading frame right here, that this is a potential gene. And so we would need to go investigate to see whether this is truly an open space without genes, or if there are some genes in here that the computer missed. So the goal for the course is to predict genes, gene starts and functions for all the genes in the 6-phase genomes: STEMI, Acolyte, Aminase, SANSA, Last Resort, and hopefully eventually Eden when we get the sequence. The learning goals for the 3 videos that come in 3 different parts is to learn what the basic principles of annotation are, and that's what this video will cover. But we'll also learn how to collect and analyze data that help us make good decisions about open reading frames and starts. We'll learn how to record the data and analyze the data and log our thought process for making decisions about gene calls in an electronic notebook. And we'll also learn how to present your gene calls supported by evidence to your peers, and that's what you guys will be doing in class. So I want to emphasize that before you watch this video, it's really important that you do the reading assignments before watching the video. And so this is the reading assignment for the next 3 videos. You need to read the annotation guide and pay particular attention to the guiding principles of phage annotation, which is what we're about to cover now, and later on pay attention to these sections in the Mechanics of DNA Master. There's a third document that I want you to look at, which actually will be titled B&B 155 2018, How to Call a Gene and Document Your Work. And that is a sample electronic notebook entry that I want you to use as a model for your own notebooks when calling genes. So the first principle tells us that any segment of DNA, so any region of sequence, usually only one frame in one strand, so only one frame out of the 6 frames, has a gene in it. So each double-stranded segment of DNA is generally part of only one gene. So here again, we're looking at the 6 different frames. Here's gene 2 of SANSA, here's gene 3, but there's also a reverse gene, number 4 here. Can you see that this is the same region of DNA, and yet there's 2 genes called here? And so based on this principle, we would predict that probably only one of these genes is correct. And so we'd have to analyze the data for both of these genes to decide which one is more likely to be the correct gene. So I'm predicting that it's probably going to be the reverse gene, number 4, that's not a real gene, and I'll talk a little bit later about why I think that's true. So guiding principles 2 and 3, genes do not overlap by more than a few base pairs, although up to about 30 base pairs is acceptable. Typically I see 8 to 12 base pair overlaps, but if I can find a smaller overlap or a really small gap, that's usually preferable. The gene density in phage genomes is very, very high, so they tend to be tightly packed. That means large gaps are not usually acceptable unless there's some sort of regulatory sequence there. So we often try to choose the longest possible open reading frame without having a huge overlap. So here I'm showing you an example of GP49 and GP50. And if you look closely here, you can see here's the start chosen for GP49, and here's the stop for GP50. I'm looking for, if I'm analyzing GP49, I'm looking to analyze the overlap with the stop that's just upstream from GP49. Here are the coordinates for GP49 start and the coordinates for GP50. So I can calculate that by saying which nucleotides are shared by both GP50 and GP49. And so if you take the higher number, which is 34,854, and subtract the nucleotides that are not included, so that would start at 34,850, then the answer is a 4 base pair overlap. Now 4 base pair overlaps, which we'll talk about in more detail later, are a sign from the annotation guides that you probably have found the correct start. 4 base pair overlaps are highly favorable, especially for genes that appear to be in the same operon. So here's a situation where there's a gap between GP47 and 48, and so you can see that the second start has been chosen for 47 rather than the first start. So here's a situation where I would expect this gene to come all the way up to the stop codon of 48, and so I would look very closely at this other start to see if it is if the evidence would support this start over this one. So clearly the Glimmer and GeneMark programs chose this one, but this is a situation where I'm willing to bet that Glimmer and GeneMark are probably wrong. So the start that creates the longest open reading frame and the shortest gap is usually the favored start. That's not always true, but it's most often true. The fourth guiding principle tells us that most protein coding genes will have coding potential predicted by Glimmer, GeneMark S, or a host version of GeneMark, so GeneMark M-SMEG or GeneMark Gordonia. And start sites, not only does the gene have to have coding potential, but the start sites are chosen, and this is really important, to include all the coding potential. So let's look at our example of GP3 and 4. Which of these two genes is going to be the more likely gene? Is 3 not a gene, or is 4 not a gene? If we look at the coding potential in this region, and here again we're looking at the 6 different frames in this region of the genome. Right here you can see this huge spike in coding potential, and these are the coordinates for GP3. And there are 2 starts here for GP3, and then you can follow along the open reading frame, and here's this downward tick indicating the stop codon for GP3. So number 1, this gene has very high coding potential. Number 2, I'm noting that there are 2 potential starts for this gene, and either one could be appropriate given that they both include all the coding potential. If you look at the 3rd and 4th start, I would never choose those because they do not contain all the coding potential. If you look down here in this first frame of the reverse genes, you can see here's the stop for GP4, and here's a number of different starts. The first thing you're going to notice is there's virtually no coding potential here. So if I had to choose between these 2 genes, clearly I would choose GP3 as being real, and GP4 is probably not a gene because it doesn't have coding potential. Oh, and here they're indicating where those genes are. All right. Many phage genes are unique and will not have any homologs in the database. So every time we isolate a new genome, it's an opportunity to learn about new genes that nobody's ever seen before. So sometimes the rule is that something's more likely a gene if it matches a gene in the database. But when we're looking at phage genomes that are highly mosaic, we frequently come across genes that don't yet exist in any of the databases. And an example is here in this Famarator image, where we have 3 forward genes here, and this white box indicates a gene that doesn't match any gene in NCBI database or the PhasesDB database. And so long as this gene is long enough that it's more than 120 base pairs and has coding potential, and maybe even if you plug it into HHPRED, which you'll learn about shortly, you can see that it might have a predictable function. There's all kinds of other things that we can look at to indicate whether it's a gene, even though it doesn't match anything in the database. So here's the coding potential for that gene. We can see that it has coding potential. Some protein coding genes may not be predicted by Glimmer or GeneMark. Therefore, all the open reading frames that are over 120 base pairs that fall into gaps in predicted genes in the annotation should be carefully evaluated for similarity to genes in the database. So here's again this gap between GP37 and 38 that I showed earlier. I would probably figure out what the protein sequence would be for this open reading frame, and maybe even this little guy right here, and blast those to see if it matches anything in the database. I would also check to see if these had coding potential. If there are 2 genes transcribed in opposite directions, we talked about this earlier, then we have a region of divergent transcription. So these 3 forward genes would need a promoter, and this reverse gene would need a promoter. And that means when you have these regions of divergent transcription, you don't want overlap. You actually need a gap between these 2 genes so that there's room for a forward promoter and a reverse promoter. So here's the example that we used before with GP59 and 60 in SANSA, where this gap is actually appropriate. Okay, protein coding genes are generally at least 120 base pairs. That means they have 40 codons, or 40 amino acids long. There are examples of proteins that are as small as 26 amino acids that are functional. But in general, we're going to be suspicious of any open reading frame that's less than 120 base pairs and look really hard for evidence that would convince us that it is actually a gene. Switches in gene orientation from forward to reverse or vice versa are pretty rare. So here's a group, for example, genes 22, 23, 24, 25. You can see that they're all practically end to end. Maybe they even have that 4 base pair overlap. These are genes that are likely part of one operon. It would be really weird to see all of a sudden a change in orientation of genes, like a reverse gene somewhere in this region. Likewise, you can see all these reverse genes going in this direction, and it would be weird to see one single forward gene in the midst of all these reverse genes. Each protein coding gene ends with a stop codon, and these are the 3 flavors of stop codons that we learned about last semester. And of course, they have to have a start codon as well. And what you may not realize is that there's more than one type of start codon. So last semester, we really focused on talking about ATG starts, but in bacteria, open reading frames sometimes start with a GTG or a TTG. Now, the ATG and the GTG are the starts that we see most frequently, but every once in a while we see one of these TTG starts. Upstream is language that Keith Melody and I will use very, very frequently. So I want to make sure you understand what we mean by upstream. So here are 3 genes that are forward genes. And so when we're talking about upstream, we're talking about the flow of transcription. So if you can think about the flow of transcription like a stream, then any sequence that is before the coding regions of genes, that's termed as upstream the flow of transcription. So this region right here is upstream of GP20, 21, and 22. If we look at this reverse gene down here, this region of the genome is considered upstream of GP19. So here's our example of overlaps again, and understanding what upstream and downstream mean will be important for trying to calculate what the overlaps of genes are. So one of the things that comes into play when you're making decisions about where your start codons are for genes is being able to look at not one piece of evidence, but multiple pieces of evidence, and deciding which piece of evidence to weigh more heavily in making your decision. And so one of the most important pieces of evidence to weigh is the location of your start codon relative to the stop codon of the upstream gene. So over here in the corner, I'm showing these 2 reverse genes. Okay, GP49. Here's the direction of transcription. Here's the start of 49. I'm going to determine how many nucleotides are shared by the 2. And so in this case, it's one of those fantastic 4 base pair overlaps. I am going to be pretty certain that that is the correct start for this gene. I have to check all the other evidence, but this is a very strong piece of evidence that's going to probably trump other pieces of evidence. For example, if this start has, doesn't have the best Scheindel-Garno sequence of all the possible starts, the fact that it's a 4 base pair overlap outweighs the poorer ribosome binding or Scheindel-Garno sequence score. Okay. And so here I'm indicating that this evidence outweighs a poor Scheindel-Garno score. Okay. This is a whole lot of text. This comes down to more ideas about how to weigh your evidence and figure out which piece of evidence to rely on in order to make your decision. Another thing to keep in mind is that starts for genes are usually conserved in other versions of that gene in other genomes. And so we call those other versions of that gene homologs. So what we look for is to see whether or not genes have the same start, and then did other people call the same start. Let's give an example of this. We can use BLAST data to help us determine how our protein aligns with proteins in the database, and if those proteins in the database have the same start and were called in the same way, then the first amino acid of our query protein sequence, which is the amino acid sequence of the protein in our genome sequence that we're analyzing, does it match the same for the first amino acid of the target sequence? So whatever it matches up with in PhasES-DB or NCBI, do they start in the exact same place? And you can see in this particular example, they do, MTA, MTA. So in this sense, the query, the first amino acid of the query protein, our protein, matches that with what the amino acid sequence that it matches in PhasES-DB. So Q1 equals S1. And this is the notation we will use when we see these kind of alignments. Another way that we can check to see whether all the homologs of this particular protein sequence are shared is through something called Start-Orator, which is part of a program called Pecan that you guys are going to learn about in a couple of videos. So each one of these pink tracks shows a homolog of the protein that we're looking at. The protein we might be looking at is in track 3, and here all these lines indicate the potential starts within that protein sequence. These other tracks are showing the homologs, and you can see that start number 1 of our protein is not found in some of the tracks, but is found in others. So start number 1, even though it gives the longest possible ORF, is probably not the start because it's not conserved in all the homologs. And so the rule here is that often the start is conserved. What we can see is the start that has been chosen for a protein, number 2, has also been chosen in all of these other homologs with the exception of track number 11. And so the most annotated start is start number 2. And so that's another factor that we weigh in making our decision. Is the start conserved, and is it also the start that most other people chose when calling the starts of the homologs? So no one piece of evidence alone should be used to make decisions about a likely start position. You have to weigh all of the evidence together. Sometimes all the evidence will point to one start, but often you'll see some evidence pointing to one start and other evidence pointing to another start. And so you have to make the most reasonable call based on the evidence that you have at hand. So we might have a start that includes all the coding potential, is the most annotated start in Start-Orator, and maybe it has the best Scheindel-Garno sequence score. But it also might create a 30 base pair overlap, which is less favorable. But all these things, and maybe all these things outweigh the fact that it creates the less favorable 30 base pair overlap. So in our next video, I'm going to show you how to collect data, analyze data, and record all the evidence and the logic of your decision in your notebook.